So my laboratory is interested in sort of unraveling the genetic underpinnings of malignant glioma and glioblastoma in particular. And in the first half, and we are in particular interested in identifying those genes that play a critical role in regulating therapy responses of this tumor type. So in the first part of my presentation, I'm going to talk a little bit about a novel BCL2 family protein that we have identified, which is called BCL2-like-12. And I'll tell you a little bit about how we think this gene functions, how it contributes to the pathogenesis of the disease, and how we think it plays a role in regulating therapy susceptibility of this cancer. And I will also tell you a little bit about how we think this gene is regulated. So I will talk a little bit about microRNAs, in particular microRNA 182, and how we believe this microRNA can regulate L12 expression. And then in the second part of my presentation, I'm going to talk a little bit more about nanotechnology, how we think we can implement this technology into our research program, and how we can leverage some of the genetic findings and the cell and animal models that we have generated to actually drive certain nanotechnological platforms, in particular RNAi-based platforms, towards clinical application. So kind of the mission statement of the lab is to define as well as to overcome therapy resistance of malignant brain cancers. So like many other labs, my lab is also part of the Northwestern CCNE, and really the overarching goal of this center grant is to sort of integrate many different areas, bring together many different scientists with many different expertises. So we try to bridge cancer genetics, cancer biology, so the identification of novel targets, the characterization of these genes in both cell culture as well as in mouse models, and then kind of to triangulate this with nanotechnology development to eventually come up with novel leads, novel directives in order to treat this disease. So if we talk about malignant brain and CNS cancers and glioblastoma or GBM in particular, we're talking about a highly lethal, incurable cancer with one of the poorest 5-year survival rates. So we're talking about a disease that has a very progressive course, and typically patients die 16 to 18 months after diagnosis. And unfortunately, this prognosis really has not really changed significantly over the past couple of decades. And the problems or the challenges that we are facing in the glioma field, they are staggering. And on this slide, I've listed probably the two most important ones. First and foremost, we have to think about therapy delivery, because the brain tumor is a very isolated neo-organ. It is really separated from the rest of the body, from the body circulation. By what we call the blood-brain, the blood-CSF, or the blood-tumor barriers. And these are very robust biological barriers that can prevent the accumulation of therapeutic drug concentrations within intercerebral tumor sites. So one emerging question here is, can we develop novel delivery platforms that more effectively and perhaps more selectively help drug cross this biological barrier? And then the second problem is that we have to understand the genomic, genetic, and post-translational complexity of the disease. So we not only have to look at isolated oncogenes, have to understand how this isolated oncogene functions, but we have to understand oncogene function in the context or against the backdrop of entire oncogenic or tumor suppressive signatures. And so one emerging question here, of course, is, can we develop combinatorial regimens that not only target isolated oncogenes, but entire oncogenic signatures? Why is this important? Because GBM, like many other solid cancers, is characterized by a very high degree of genomic instability. So this is a typical array CGH profile derived from a GBM patient. And you can clearly appreciate that this patient here has a very robust gain in EGFR, the epidermal growth factor receptor, which is a signature oncogene of GBM as well as other cancers. But you can also appreciate that across the entire genome, there are myriad additional genomic aberration, multiple gains and losses across the entire genome, one of which targets the BCL12 oncogene, which I will talk to you about in a second. So it is therefore not completely surprising that if we take patients, even those that are stratified based on their EGFR status, and treat them with an EGFR inhibitor as a monotherapeutic approach, that these therapies usually fail. Usually we can identify 5 to 10% of responders, but tumor recurrence is almost always universal. So let me tell you a little bit about this gene that we have identified a while back and that we kind of used as a kind of proof of concept model system to evaluate nanomaterials, RNAi-based nanomaterials. So I pointed out to you it resides on chromosome 19q13, a region that has been implicated in increased radio and chemotherapy resistance. Initially, we did very detailed expression analysis, both on mRNA as well as on protein levels. And basically what we found was that BCL1212 is significantly overexpressed in glioblastoma multiforme, so in grade 4 malignant astrocytomas, with very low expression in normal brain, and low-grade astrocytoma, where it is confined to endothelial cells or very weakly to neurons. We did low-throughput transformation assays of BCL1212, as well as adjacent genes that localized to the same amplicon, and we could see that L12 seems to have some sort of cancer-relevant activity. So what is BCL1212? It is a very atypical member of the BCL2 protein family. It has what we call a BH2 domain, a BCL2 homology domain 2, and this domain shares significant homology to the BH2 domain of other members of the family, in particular canonical members such as BCL2 or BCLXL. But this is pretty much where the homology begins, and that's also where it ends. It doesn't have multiple BH domains, which are critically important for anti-apoptotic members of this family to interact with pro-apoptotic ones and actually block their activity. And consequently, in phylogenetic analyses, L12 really doesn't cluster with the rest of the family. So structurally speaking, L12 is sort of the black sheep of the family. And we spent quite a few years to also characterize and find out how it functions on molecular and cellular levels. And it turns out that also functionally, L12 is the black sheep of the family. So we found that L12 doesn't really safeguard mitochondrial membrane integrity, so it does not prevent the release of cytochrome C from the mitochondrial intermembrane space into the cytosol like BCL2 or BCLXL do. But instead, we found that L12 is a very potent inhibitor of effector caspase activation. We found it can block caspase-7 activation through an indirect mechanism through alpha-V crystalline upregulation. It can block caspase-3 upregulation. And interestingly, we also found that L12 not only resides in the cytoplasm, but also in the cell nucleus. And here we found that it can physically interact with p53, and it can block the transactivational potential of p53. And it does this through two different mechanisms. First, it can bind p53 and can prevent p53 from reaching its target gene promoters, such as p21, and it can also decrease the protein stability of p53. Not sure how this functions. But so far we have uncovered these two activities of BCL1212 in the cell nucleus. So we of course became a little bit more interested in the BCL1212 p53 signaling axis simply because p53 is such an important tumor suppressor, not only in GBM but in cancer in general. So in addition to a lot of functional evidence, biological evidence that we had, we also thought to have some genetic evidence. So we asked, so if it's really true that L12 is an inhibitor of p53, of wild-type p53, then we would expect that L12 is expressed and gained at higher levels in those tumors that actually have p53 wild-type status. And that's precisely what we found. So we looked again in the Cancer Genome Atlas, which actually started off with malignant gliomas with glioblastoma multiforme. And we separated these tumors into those tumors that have mutant and those that have wild-type p53 alleles. And if we look at copy number alteration in this locus, we see those alterations are significantly enhanced in p53 wild-type tumors over those that have mutant alleles. And the same is true for BCL1212 mRNA, again, higher expression in those tumors that have p53 pathway inactivation in comparison to those that have a mutant p53. And we also looked across the different subtypes of GBM tumors. And while L12 is really overexpressed at quite significant levels in all tumors, the levels of overexpression do vary, and we found that L12 is significantly expressed at lower levels in the proneural subtype, and gratifyingly, this is actually the tumor subtype that is enriched for p53 mutations. So based on these findings, we then of course became interested in finding out how is BCL1212 regulated, and giving the importance of microRNAs in regulating cancer gene expression, we again looked into the TCGA data set and asked what microRNA negatively correlates with the expression of BCL1212. And one of the top scoring microRNAs that we identified was Mi182. And interestingly enough, specifically in the proneural tumor, we saw the most dramatic negative correlation between Mi182 abundance and the abundance of BCL1212 mRNA. So we went ahead and evaluated whether Me182 indeed can target B-cell12. And when we introduced Me182 in different glioma cell lines, we see a reduction in B-cell12 protein levels as well as on mRNA levels. We could identify the Me182 binding sites within the 3' UTR region of B-cell12. If we take this region here, co-expressed with Me182, we see a repression in the reported activity, and if we mutate this binding site here, this effect is gone. And it appears that this effect is specific for Me182, but other MERs that localize in the same microRNA cluster do not show this effect. Functionally, what happens if we introduce Me182 into cells? I mentioned to you AL12 is a caspase inhibitor, and we could show that if we introduce Me182 into glioma cells, co-treat with a chemotherapeutic drug, staurosporin in this case, we can see a significant increase in overall caspase activation, very similar to what we see with siRNAs targeting B-cell12. We did a bunch of rescue experiments, and we can actually show that AL12 is a very important anti-apoptotic gene by which Me182 actually exerts its pro-apoptotic effects. We also went ahead and could show that not only Me182 sensitizes cells not only to staurosporin, but also to more clinically relevant treatment regimens, temozolomide, radiation, as well as RDK inhibitors. So then, of course, the question for us became, how can we sort of leverage these genetic insights? And I think we can all agree in the cancer research community that we made really tremendous progress in many different areas. We made significant progress in the omic analyses, in particular the Cancer Genome Atlas project really accelerated these advances. We made significant progress in genome scale functional analyses, so high throughput gain and loss of function assays, RNAi screens, cDNA complementation screens, and the triangulation of these data, of these screens with these large-scale data sets really helped us identify novel critical cancer genes. And last but not least, the advances in generating more sophisticated mouse models, both orthotopic models as well as genetically engineered models, clearly helped us understand better cancer gene function in vivo. So we have a pretty good, however still incomplete understanding of really what the critical oncogenic signatures are. But the question, of course, becomes, how do we target them? And unfortunately, the vast majority of cancer genes that we have identified and probably will continue to identify are what we call undruggable. And I also want to make the argument that AL12 and its targeting microRNA fall under that category. So the question really becomes, what are we going to do with these genes? And of course, a very logical answer to the question is RNA interference. But we all know it is very difficult to robustly and safely deliver RNA to tumor tissue. And that is pretty much the problem that we try to address, and that's why we teamed up with Chad Merkin's laboratory on the Everson campus, and I think Teri gave a very nice introduction of the technology. So basically, we are working with what we call spherical nucleic acids, or short SNAs. So we're looking at a gold nanoparticle that is pegylated for stability purposes. It has a diameter of 30 nanometers. And the Merkin lab was able to functionalize these particles with a small double-stranded RNA. The chemistry is not too complicated. Basically, the antisense strand is modified with an alkyl spacer, and then the duplex is conjugated to the gold through a gold thiol bond. And these particles have a bunch of really interesting features. First and foremost, we're looking at an agent that is capable of transfection and gene regulation at the same time. So we simply can take these particles, add them to cells or tissue. No auxiliary transfection agents are required, no viruses are required. In addition, these particles are very stable in physiological environments. They are able to enter a wide variety of different cell types as well as tissue, do not exhibit apparent toxicity, and induce less activation of the innate immune response. So we went then ahead and tried to evaluate this platform as a potential novel anti-GBM therapeutic. So in the next couple of slides, I'm going to touch upon four different areas. I'm going to talk to you a little bit about the ability of these particles to very effectively penetrate cells. I will talk a little bit about efficacy and functionality of SNAs that target BCL1212, so siRNA-based SNAs, and also Mi-182-based SNAs. I will describe to you how these particles are able to knock down BCL1212 and how this knockdown of L12 impacts downstream signaling surrogates of L12. These are effector caspases and p53. And then I'll talk about the delivery of these particles, and last but not least, I'll show you some data that we had on in vivo efficacy of these particles. So as I pointed out, these particles penetrate cells very effectively. So in the first panel here, you can see you had 7 cells with a transformed human glioma cell line, patient-derived tumor neurospheres, as well as mouse model-derived tumor neurospheres. And in all cases, you can see within a few hours, these are Cy5 labeled SNAs, where we put the Cy5 on the PEG moiety or on the RNA moiety. And you can see that these cultures are really significantly penetrated by these particles. Interestingly, this uptake seems to be dependent on the presence of scavenger receptors. The Merck lab did some more elaborate studies, and they found that scavenger receptor A seems to be a key player in the uptake of these particles. So correspondingly, if we pre-treat these cells with poly-I, we can significantly decrease the cellular uptake of these particles. Then is the knockdown that we can elicit by these SNAs functional? And the answer is yes. So we can nicely downregulate BCL12 protein levels in transformed glioma cell lines. Roughly 100 picomolar of these particles appear to be sufficient. We can do this also in patient-derived tumor neurospheres, which are very difficult to transfect and infect, and that corresponds to roughly 150 to 100 nanomolar of lipoplex-delivered siRNA oligonucleotides. We can also demonstrate by race PCR that this downregulation is actually mediated by RNAi, which is an important control experiment. And also equally important, the functional readouts that we did and that we found, it very closely mirrors what we see with siRNA oligonucleotides or with the microRNA. Again, if we take out BCL1212, co-treat with a chemotherapeutic drug as an inhibitor of effector caspase activation, we see an increase in overall caspase-3 and caspase-7 activation. It also modulates p53 activity, so expectedly, if we take out L12 with these SNAs, we see an increase in p53 protein abundance, an increase in p21, both on protein as well as on mRNA level. So these particles can penetrate cells very effectively, and they do what they're supposed to do. And then, of course, delivery. And there are kind of two aspects to the problem. First, once they are in the brain, do they have the capacity to actually infiltrate tumor tissue effectively? Do they have the capacity to pervasively go and penetrate the tumor material? And to answer this question, we did an experiment where we teamed up with also Tom Meady's lab, and he and Chad Merkin, they developed a technology where we not only can functionalize these gold nanoparticles with DNA, but where we can functionalize the DNA also with gadolinium. So we also have basically an imaging agent at hand. And in this experiment here, we inoculated tumor cells into SCID animals. After 3 to 4 weeks, we see these tumor masses forming. And then we take these gadolinium functionalized SNAs, inject them into the tumor bed, and then subject these animals to MRI. And you can see there's a very pervasive infiltration of the tumor tissue. We can also visualize this by laser ablation ICP-MS, so mass spectrometry approach, where we can quantify gold or gadolinium. And again, we see a very good co-localization of the gadolinium or the gold within the tumor. And then we can also do a 3-dimensional reconstruction of these MR images. And in red, we can see really the extent of tumor penetration that we get with these particles. And that to us was quite a remarkable finding. But then, of course, the question is, can we deliver these particles systemically? And the answer is yes, we can. So in this experiment here, we took these SNAs and inject them intravenously into the bloodstream of glioma-bearing animals. And then we isolated the tumors, and we simply looked at gold content by histology as well as by ICP-MS, by mass spectrometry. And what we found is that we see a very significant accumulation of these particles within tumor elements. And here, if you can look at a slightly higher magnification, here in brown, these are the blood vessels. These are CD31 positive blood vessels. And the speckle staining here clearly demonstrate that these particles get out of the vessels and actually have the capacity to penetrate and invade the tumor parenchyma. We did also detailed quantification and biodistribution analysis, and what we found was that, of course, the majority of these particles, they end up in the liver and the spleen, but up to 1% of the total amount injected is actually able to cross the blood-brain barrier and then selectively accumulate within tumor elements. We also did detailed toxicology studies, and we could not find any significant adverse side effects. We did detailed cytokine profiling to see whether or not these particles can induce some sort of innate immune response. And again, we did not see any significant changes in cytokine expression, in particular those that regulate the innate immune response. And then last but not least, we did in vivo studies where again we inject these particles systemically through IV injection, through tail vein injection into glioma-bearing animals. We could identify one construct that very significantly downregulate BCL12 expression specifically within the isolated intracranial tumor. And if we treat animals with these particles, we see a roughly 3- to 5-fold reduction in overall tumor burden. And if we use a more invasively growing tumor nursery-derived model system, we see a roughly 20% increase in overall survival, telling us that these particles have efficacy in vivo. Also histologically, L12 again is a anti-apoptotic gene, so if we take it out and then we look within the tumor for both active Caspase-3 as well as tunnel staining, we can see that the presence of the SNAs and associated with this, the knockdown of BCL12 within the tumor can increase active Caspase-3 activation and can increase double DNA strand breaks. We can do pretty much the same set of experiments, not only with siRNA-based SNAs, but we can also do this with Mi-182-based SNAs. The chemistry and the coupling is very similar chemistry. And when we take these Mi-182-based SNAs in cell culture, we can detect a very significant downregulation of BCL12 mRNA, and here is shown the protein levels. And also expectedly, if we treat cells with Mi-182 SNAs, co-treat with a chemotherapeutic drug against taurosporin, temozolomide, or RDK inhibitors, we see an increase in Caspase activation. And very similar to the siRNA particles that we have, these Mi-182-based SNAs have the capacity to cross the blood-brain barrier and actually accumulate within the brain and within the brain tumor. And again, there appears to be a very sort of selective uptake of these particles within tumor elements, not so much in normal elements, and we believe this is due to what we call EPR, or enhanced permeability and retention effect, which simply means that the vessel within the tumor, they are abnormal, they are leaky, they are distorted, and that kind of makes the tumor like a sponge that can actually suck up these nanoparticles very effectively. And in literature, people usually refer to this phenomenon also as passive targeting. And then last but not least, we did the first round of in vivo experiments, very similar to what we did with the siRNA-based SNAs. Again, we see a significant reduction in overall tumor burden, as well as an increase in overall survival, similar to what we see with lentiviral delivery of Mi-182s directly to cells and then inject these cells orthotopically into mice. And with that, I'm gonna finish here. So I would like to thank the people who were involved in the work. So in my lab, Sam and Fotini, they spearheaded the siRNA as well as the Mi-182 project. And many thanks also to David, Caroline, and Emily in Chad Merkin's lab, who did the imaging studies and helped us with the quantification of SNAs in tumor elements, and also helped out with the in vivo studies. Thank you very much.
Were the effects of manipulating MIR 182 consistent numerically with working on the BCL2 L12 directly?
Sorry, can you
in other words, was the effect of MIR 182 consistent with its effects on BCL2 L12?
So Mi-182 has, of course, many different targets, and we try to really catalog these targets, in particular in patient-derived tumor neurospheres. And what we found is that, of course, it regulates other critically important oncogenes, c-MET, for example, HIF2 alpha, SOX2. So it also plays roles in mitogenic signaling as well as differentiation of tumor neurospheres. But when it comes to regulating sensitivity towards chemotherapy, it appears that BCL12 is the major target and to which MI182 can actually sensitize cells towards therapies.